Okay, let's begin. I'm Mike Douglas, and this is the New Technologies in Mathematics seminar series of the CMSA. And this week we're delighted to have Eric Mjolsness from the UC Irvine Departments of Computer Science and Mathematics. Eric is a pioneer in systems biology and especially in computational modeling of biological systems. And today he'll tell us how AI is expanding the possibilities for biology and modeling. Eric.
Great, thank you. So hopefully you can see the slide at least as well as we saw it in testing it out before. And the particular ways that I think AI may be able to help modeling in complex biological systems, of course it's mathematical modeling, is by model reduction using machine learning, by spatial graph dynamics, which I'll talk about a little bit, and then more loosely in approaches to symbolic approaches to computational mathematics, computer algebra, and automatic theorem verification. Let's see. So there's a long abstract saying just that. And so jumping to the first category there, the machine learning for model reduction, incorporating various things that you know. And we have developed two approaches, but the first one is called dynamic Boltzmann distributions, and it's applied to stochastic reaction diffusion systems. The lead is being taken by Oliver Ernst as a PhD student at Salk and UCSD in Terry Sanoski's lab there. That's a collaboration that's been going on for a while. And the aim is in neurobiology to understand what's happening in a synapse. The general framework we like to talk about is if you have a fine scale system running along the bottom of this diagram and you want a reduced model running along the top that does the same thing over an interval delta t, you have to define what the same thing is. It should be agreement on some set of observables. Depending on what the observables are, that'll tell you how closely you can approximate the fine scale with the coarse scale. But basically, you want a semantics and a reduction map to almost commute. And in order to do that, you can have different kinds of mathematical models in biology. Dynamical particles, dynamical fields, such as stress continuum models, and I'll argue dynamical graphs. So we'd previously done something like this for reaction diffusion models in a synapse with the sort of central equation shown with a coarse scale probability distribution that tells you what is happening to your reduced model R as a function of time by making it a Boltzmann distribution by assumption with some set of interesting interactions, potential energies V, but the coefficients, the coupling constants mu in this easing model like a thing would vary with time rather than being constant in such a way as to approximate a much more detailed fine scale system. And in this case, the coarse scale reduced model was not spatial. And so I'll leave that one aside and say we're ready for the spatial generalization, which is the lower Boltzmann distribution equation, approximating probability distribution as a function of all the numbers of particles that you have, as a function of all their positions x and their types alpha, and the time t is again going to be a Boltzmann distribution with some one particle potential functions that vary in time, two particle potential functions, and maybe even higher, but not too high, up to capital K. And our job is to figure out what's the dynamics for nu that would let the resulting distribution P tilde look a lot like the fine scale distribution, which is the solution of the master equation, given at the top there, dPdt equals WT, and you can just describe all of stochastic biological modeling with that equation if you put the right W's in. So we want some kind of objective function that says P tilde should well approximate T. The typical thing to use is the KL divergence, so we decided to start there. And vary the KL divergence, the variational problem is ds d something equals 0. What's the something? The something is the right-hand side of equation 13 there that tells you how the potentials should evolve. It's an unknown functional. So, so S becomes a functional of a functional, and that's a sort of higher order variational calculus problem, which we'll try to get rid of by putting in parameters for everything. But in principle, that's the machine learning problem, is to learn the Fs so that you get a good approximation in the probability distributions. Well, if we just pretend that we can do variational calculus as if it weren't higher order, then you know, we can use the chain rule in the way indicated, and the derivative of S with respect to the news give you differences of moments between you know, where all the particles are basically in the fine scale underlying distribution P and the coarse scale modeling distribution P tilde. So if that were 0, if equation 20 were 0, then you'd be done. And there are some of the moments that you might get. And for the second part, we need to somehow parameterize that functional, and we've tried a lot of things. Various different things work. So again, here's our PDE constrained optimization problem. You know, find F. It's aided a bit in our way of thinking of things by this proposition that says that the master equation was a sum of operators for different processes. Every chemical reaction has its own W. The equation is linear because it's incredibly high dimensional, like the Schrodinger equation. But it's linear in P, but it's also linear in R in the processes. More processes just add in to the operator. And because we're doing this maximum entropy optimum inverse problem to get F, well, to get nu really, then you can see that you can get the same linearity for the Fs, for the unknown F. So F should decompose into a sum of processes. And that means that we could take simplified models or even analytically solvable models for some of the sub-processes and use those as sort of basis functions. And that's what we do. So here, for example, is a little chemical reaction network, stochastic reaction diffusion network, where you have fratricide or mutual annihilation of particles and replication and you know, decay and so on, and also diffusion. And we assume a lattice for the spatial state variable at any time, so we discretize it onto a lattice, and we want to know how the H's and the coupling constants J ought to evolve. And the idea is to solve the separate. These are all very simple chemical reaction models. They're kind of solvable if you didn't have space. If you did have space, then you can use the you know, 1D solution of the Ising model to crunch through. And we did that using Mathematica to compute some of these basis functions that you see over on the right side in very tiny print because they get big. And so that's our guess for you know, what the basis function should be, and then we try to combine them in various ways in a sort of neural network architecture, so as to get a good dynamical system for H and J that well approximates a stochastic simulation, again on a lattice of the same system.
Eric,
yeah,
I'm sorry, I got somehow a little lost here. So the underlying system is a chemical reaction network here, or
yes, stochastic chemical reaction and diffusion. So we have diffusion.
Okay, so there is time and space.
Yes, right.
And now the Z, the equation you gave here is going to be a model for that. It's going to be
Z is the partition function of the probability of the instantaneous probability distribution of the model of the coarse scale reduced model.
And the S's are amounts of.
The S's are spins. They're just Boolean variables that say whether my lattice space site is occupied by a molecule of type A or not.
Okay, so that was already present in the underlying model. A site can be occupied with a molecule or not.
Right.
And,
right. And
but there's a dynamics, and now we're fitting functions HJ that describe the average behavior under this dynamics.
Well, what you're fitting is the dynamics for H and J.
Yeah.
So H and J, then you initialize it to be accurate, and then from then on, you're trying to create a law for the evolution of H and J that tracks.
So it's a non-equilibrium.
Yes, right. Intrinsically non-equilibrium.
Okay, so then the H and J evolve. Okay, I start to see. Okay.
Yeah. And at the bottom, you see the learning process of mean and nearest neighbor correlations, and well, it learns. So it can learn to track this system, and you get you build a lot in with these well-chosen basis functions that are illustrated there by using the computer algebra to crunch through and get a good basis set. So the resulting algorithm is a variant of the Boltzmann machine learning algorithm of the 1980s, which Terry Sinofsky was a co-author on. You have a wake phase and a sleep phase. That's where you measure those two different kinds of moments. If they're the same, you're done. You have a forward propagation phase where you solve the PDE. You have a backward propagation where you compute gradients of what happens later in the system as a function of what the parameter values were earlier in order to add that all up. And there are a few little wrinkles that are not on this slide exactly, including sort of a moving window that are described in the paper. But this is a PDE constrained optimization algorithm that looks like a mixture of an adjoint optimization algorithm and the Boltzmann machine learning algorithm. You can introduce hidden variables in the easing model, and that tends to make things better. So there's a couple of different topologies for how you could do that, and the corresponding easing model expressions. And the numerical result on the resulting F's, the right-hand side of the PDE for where the particles are, is much improved by having the hidden units, which of course is consistent with what happens in neural networks generally. We can even do a 3D that it takes some more work to not be using an analytic solution of the easing model in one dimensions, but use some kind of finite element method in 3D that's described in the paper. And you can get sort of track with the reduced model these waves of activation that roll through the Rossler oscillator in 3 dimensions. And after a little while, it loses track. It kind of decoheres. But for the first couple of oscillations, then it can track the actual system. Okay, so that's and we're continuing to develop that by applying it to calcium processing reaction networks in neurobiology. So that's the next goal there. Now, another model reduction adventure that's pretty recent is with my PhD student, Corey Scott at Irvine. And he's invented something he called graph prolongation convolutional networks. I'll say what that is for biomechanical model reduction in microtubules. But the framework is that we'd like to have a multi-scale way of looking at graphs. And you know, you can have sequences, regular families of graphs that grow in complexity from one generation to the next. You can link them. We call that a hierarchical graph sequence. You can link across the generations to get a relationship between a fine scale graph given near the top of this tree and the coarse scale graph at the top to the finer scale graphs at the bottom. And if you have both of those things, then we call it a graph lineage and try to use it as an architecture for machine learning. How do you generate these regular families of graphs? You could do it with a graph grammar, and here are some possibilities for doing that. We'll come back to graph grammars later, but that's one ingredient. I mentioned the graded graph idea, where we're going to link across scale between coarse scale and fine scale versions of a graph, and we can do that with an orthogonal matrix. And this has been useful before, for example, for algebraic multigrid methods in hot field relaxation-based neural networks a long time ago. So the connection matrix of the graph gets multiplied fore and aft by an orthogonal matrix, and you want to find a good one. You can do the same thing to the graph Laplacian as opposed to just the adjacency matrix. And that's what we're going to do in order to find a distance measure between two graphs. So given two graphs, G1 and G2, the top line suggests that you should find an orthogonal matrix P, which if you first run the diffusion operator on graph 1 and then prolong to graph 2, that should be similar in Frobenius norm to first prolonging to graph 2 and then running the diffusion operator there, possibly with a different time scale. So that's what the alpha does. It lets you say, well, graph 2 is bigger, so we'll let diffusion take longer and put in a time dilation factor there. But you can optimize over that as well. And so this is something that is a little bit hard to compute. It's quadratic with quadratic constraints and an outer optimization over alpha. And Corey worked pretty hard and got the best algorithm that we know for computing this thing. One could ask for more. One could ask P should be sparse and so on, but that might be a good thing, but we can't solve that problem yet. There's an archive preprint on this computation. So then the idea was to take this very structured microtubule molecule that's important for a lot of our biological applications, including some I'll talk about in a bit, and try to understand its biomechanics. If you pull on it, if you clamp one end and then pull down on it on the other end and it bends, how much bending and how much energy, deformation energy is there in each of the many tubulin proteins in the microtubule. So you can see the little colored picture there that shows blue along the sides where there isn't much stress energy, but there's stretching at the top and compression at the bottom. And so those tubulins get a little hot looking. And so how could this be a machine learning problem? Well, the ingredients are to start with a fine scale graph of the believed structure of microtubule when there's no defects. And that is the densest of those tubes on the lower left part of this slide, and then repeatedly coarsen it in such a way as to minimize the distance that I showed you in the previous slide. And so those are kind of the winning structures. Corey tried a lot of structures, and those are the winning structures for preserving local flow of information on that graph.
Eric, could you just say simply what's the microscopic model? Is it just like a mechanical ball and spring? Or
yeah, I was about to say that.
Okay.
It is a mechanical. So basically, there are sticks between those balls, yeah. And so at a bare minimum, there are pairwise spring energies, yeah. But there's also triples, and the triples depend on which microtubule it is exactly, whether it's an acute or an obtuse angle. It's not quite rectangular. And so there's 4 or 5 coupling constants, and it's all run in the LAMMPS molecular dynamics simulation. And this is not a very well understood art, because there isn't a sort of deep literature the way there is on atomistic simulations on what these potentials ought to be. So we pick one that at a sort of unreasonably low temperature does not make the microtubule explode when you pull on it. And so just to get the sort of quote ground truth, Corey had to do some parameter optimization to get a reasonable straw man. But presumably, as we know more, we'll be able to get those potentials better. And there are examples in quantum many body simulations of, for example, using even recently FermiNet, for example, was showed to get you the nitrogen to nitrogen potential to pretty high accuracy, and then that's a classical thing that you could put into a simulation like this.
And then the reduced model is the same type but fewer bodies, or what's the reduced model?
So yes, the reduced model will be actually, the reduced model will not be the same type. Okay. It will be fewer potentials, fewer proteins, fewer balls, but it is in fact a graph convolutional network whose goal is to predict the energy per protein at every protein in the structure. And that's the architecture that's shown in the middle here. It works at the finest scale, has graph convolutional for that, and then it has a stack of them as you coarsen. And so the idea is that long wavelength degrees of freedom are going to be slurped up by the coarsened prolonged graph convolutional networks at the bottom of that stack. And then you add up all those effects, and in both statistical and computational cost measures, this method does much better than itself without the hierarchy, than diffpool, which is a good competing method, and a few other sort of machine learning strawman that we were able to think up. And of course, it's vastly cheaper than running the original molecular dynamics. So the idea now is you've developed a method, a model that predicts energies, and so if you move the inputs to the models, the protein centroids, then you can compute gradients, and that could be the input to your molecular dynamics simulation instead. But we haven't done that yet. But other people have done on that kind of thing on atomistic molecular dynamics simulations, ISIF and is one of the team members of a fairly big team that's done that a whole bunch. So unfortunately I don't have that citation here, but I kind of like that work. That one was trained on density functional theory. Millions of density functional theory simulations for the fine scale. So because it's atomistic, you have a lot more confidence in the ground truth. So, but this is at a much coarser scale, so you could get to much bigger simulations than you ever could, or than you could in the foreseeable future using the atomistic level. All right. So the idea then is to apply this kind of thing to our simulations of what the microtubules are doing in the outer cortex of plant cells during plant development. And I'll show you a simulation like that in a bit. So here is a now yet coarser scale model of microtubule treadmilling. And this is using a dynamical grammar model that I'll show you in a minute. And it just shows that we can use a highly scalable Department of Energy sort of aimed at exascale scientific programming package or environment called Cabana, built on Cocos, to do these kind of graph-like things. The little bursts of color are where two microtubules are running into each other and joining up. And this is the kind of dynamics that we have in the plant cell case of interest. So now I'm going to use the microtubules in plant cells to illustrate the operator algebra formulation of dynamical graph grammars as a straight up generalization of stochastic chemical reaction networks, but one which really has much more kind of computer science expressive power. The situation is this multi-scale viewpoint of microtubules. Here it is again. And you can see there are these cortical microtubules in the middle plot there, middle drawing, that line up on the inside of the outside of a plant cell and really govern where the cellulose is going to be deposited in what direction, which is what governs the biomechanical properties of those plant cells, and why the corn stands up and things like that. So, and pattern formation, where do new branches go and all that kind of thing. So the cortical microtubules have a big role to play in plant shape at all scales. And here's a kind of microscope, confocal laser scanning microscope showing a movie taken over a half hour showing the treadmilling and the joining and so on that these microtubules do. I'll just show that again. And if we zoom in on this movie a bit, then you can see at the top row, you see collision of microtubules and followed by joining up, bundling or zippering. And at the bottom, you show collision followed by catastrophic depolymerization, catastrophe. And those are both possible outcomes of what happens when one microtubule runs into another. And a third one is that they can cross over. And in the middle here is shown the grammar, graph grammar kind of way of expressing this, where you have the black circle is the growing end of a microtubule with a certain position and orientation. And it can turn into interior node and create a new end node that makes the microtubule longer. So it's kind of a spatially discrete compartment of microtubule model. And that happens with a certain propensity per unit time that depends on all kinds of things, including whether you keep on going in a straight line or not, which you tend to. Likewise, there's a retraction at a retracting end. And then the next rule after growth and retraction is a microtubule growing end running into the middle of another microtubule with three possible outcomes. It could join, making that black triangle. It could go into catastrophic depolymerization, making that black square, or it could cross over, making that black diamond. There are three different states because they will tend to have three different sets of microtubule associated proteins with them. So they're actually biologically different. What's going to happen and which one happens can depend on the angle of incidence. So we'd like to just be able to express this kind of thing, and here's a language for doing it. And I already showed you a simulation that gives you one of a number of different ways of trying to actually simulate these things. But what's the, go ahead.
Yeah, just I want to not understand in detail, but just the general concept. So this is basically describing a Markov process.
Yep, it's not quite Markov because it's continuous in time.
Okay, so it's a stochastic ODE, and then the state is you have a discrete choice of graph, and then there's like labels or,
yes.
or variables on the nodes and perhaps on the edges.
Yes.
And then you can match any of these rules with pieces of the graph.
There you go. That's it. Right.
Okay, good.
And it's continuous in time with the propensity for a discrete change to happen, which is just the way it is in a Gillespie simulation of a stochastic chemical reaction network. So, if we're back to chemical reaction networks, then all this can be expressed in the Doi-Politi formalism, where you have n particles and you can get one more by taking the generating function and multiplying it by z. You can get one less by taking your generating function and taking its derivative. Then you get the law of mass action the proper way. And those are the raising and lowering operators in an algebra. This is for pure, no graphs, just stochastic chemical reactions. And so the operator for a, for a reaction looks like a annihilation operator annihilating all the things on the left side of the rule, and then creation operator instantaneously thereafter creating all the things on the right-hand side of the rule multiplied by the propensity. And those operators have an algebra, and so therefore the operators for you can take the product of two such reaction operators and commute everything until you return to the same form and you get a sum of other such. So products of these kinds of operators turn into sums of other such chemical reaction network operators. And that's the algebra for a chemical reaction network. I would like to find out if there's something similar for graphs, and then I'd like to use it in the same way for making algorithms. So here's how you can use it for making algorithms in the chemical reaction case. You try to exponentiate that operator. You can decompose it in various ways, for example, diagonal versus off diagonal, and then you get the operator product expansion, and that lets you rederive the Gillespie stochastic simulation algorithm, and also a bunch of other algorithms that are generalizations of it for molecules that have labels or positions or things like that. So that's the goal. Now, how do we generalize from particles to graphs? Well, here's the operator for an arbitrary chemical reaction network. You kill everything on the left side, you create, even with their parameters, you create everything on the right-hand side, including all their parameters. Let's just do the same thing for a graph rewrite rule like that Treadmilling graph microtubule growth rule. You would destroy the node on the left with its parameters, and you would create the graphlet on the right with its parameters. So first kill a left graph, create a right graph, multiply that by a propensity, integrate over all the possible values of the parameters that are sort of emergent from this rule, like where the new microtubule, the new object goes, and that's the operator. What is its algebra? I think this is just showing pictorially graph in and graph out. Okay, so we're not quite ready for the algebra yet. Instead, this is showing you how it would apply to the microtubule graph grammar that I showed. There's the plus end extension rule and its operator. It's more creation than annihilation. The minus end retraction and its operator is more annihilation than creation. And if we compute the commutator, then at some labor, you can find that they essentially commute. There's some extra terms that you have to throw away that involve dangling edges in the graph that don't make sense. And if you sort of trim it, then you get back to what is simply a renumbering of the objects in the shortest possible microtubule, which just has two ends and no middles. So that commutator kind of makes sense. What about growth versus bundling? That one is a little bit more complicated. So there's the growth at the top, plus n growth, and the bundling in the middle. What happens if you switch the order of those things? You get a bunch of terms, some of which just violate syntax, so I leave them out. Some of which are unlikely. For example, the one at the bottom would result in an incredibly contorted microtubule. And then if we had other rules that impose probability based on a Boltzmann factor involving bending energy, then that would be very unlikely to happen. And then the one that's likely is the one in the middle that says, first the colliding microtubule grew and then it hit. And that's the commutator. That's the difference between the two. So, so what is it in general? Once we have this, then I think that you have kind of the foundation for building algorithms. You see we already have some algorithms, but this will be a deeper foundation. That's why I'm interested in this. And the answer is the same, of the same form as for a stochastic chemical reaction kinetics. If you follow one graph grammar rule by the other, and we're just considering the off-diagonal terms, every operator also has a diagonal operator that you subtract in order to conserve probability, and I'm ignoring those ones because they're easier. So the hard case is, you know, rule 1 fires and then does something, and then rule 2 fires and does something. And the result is a sum of other graph grammar rule operators for rules that weren't necessarily in your algebra. And it's a sum over all the possible sort of edge maximal subgraphs of the output of one graph that go into the input of the next graph. So that's this sort of graph subgraph recognition thing. And for every such output of one that could go into the next one, then there's a new combination that says what's left over after you create stuff and then immediately destroy it. What else is there? And it could be a lot of fragments. It could be an interesting new graph. There's you know, just a few little technical definitions that say what are those things? What are the two graphs in the new graph grammar rule exactly? And that's what that notation is kind of defined in this paper, which is now in the Journal Bulletin of Mathematical Biology. I should mention there's another whole approach to all of this, which doesn't compute commutators as far as I know, but it does define a semantics for graph grammars that's not given in terms of operators, and furthermore, they can find the commutator equals 0 condition, and this is the double push-out definition of graph grammar rewrite rules that is described in this nice book by Erig, Prang, and Tanzer. And there's a whole community that, a small community that looks at this kind of thing, and there's that kind of a condition involving lots and lots of category theory defined maps that say whether the thing is going to commute or not. Nice one about this is I think it's not as nice because it doesn't have the operators and you can't be as quantitative in that way, in that sense, but it's very nice in that you can generalize the kinds of maps that appear in these push-out diagrams to be other things than just the category of graphs, which is what this is in. Okay, so of course, with Corey's prolongation maps from one level of a graded graph to the next, one can consider the infinite limit, and this has been done forever, and though not in those terms. And for example, if we take compute the Green's function of the associated with the Laplacian for diffusion on a graph, on a grid graph, as shown on the left here, then the contours, when you make the grid graph big, the contours circularize. There's an intermediate range of scales in which you've basically got the continuum limit. So we ought to be able to take continuum spatial limits as well. And that's a potential way to do it. In biology, we need things like manifolds to represent idealized cell surfaces, for example, where you ignore all the bumpiness, but and idealized tissue geometries where you ignore the cellularization, but also non-manifold geometries, including cell complexes. Obviously, then we have to worry about, we have to disambiguate between biocells and geocells, but even more generally stratified spaces. And that's another way of composing manifolds into topological spaces that can handle things like these topologies of microtubules shown in the lower right and shown expanded here. Here's some of the dances that microtubules go through in 3 dimensions during plant cell division. And you can see they do not obey the rules of a cell complex. You've got these one-dimensional cables running through 3-dimensional volumes. So you really need the more general, if you're going to make a continuum model of that, which is, maybe you shouldn't, but if you want to, then you need something like stratified space with differential equations running on all the different strata and agreeing at the boundaries. Well, one way to get that is to define everything on a set of graphs and then take a graph limit. And that's kind of the goal for kind of finishing out what's then a description of a very general language for describing processes that run on objects, extended objects in biology. Well, now I'd like to take the last little bit of time to discuss the other side. We've been talking about mostly numerical, machine learning, and other numerical approaches to formulating models. You could say that the graph rewrite rules are heading towards a symbolic approach, but it's still encrusted with lots and lots of numbers, which is good. But what about straight up symbolic artificial intelligence? Does that have anything to contribute? And my claim it does by way of declarative modeling languages, declarative representation of models. So for example, the dynamical graph grammar is itself a symbolic object. It has graphs that are structural. It has algebraic expressions, which are the propensities, which is a syntax tree, and you have a collection of them, and you could even have further hierarchical structure on top of that. And so that's a symbolic structure that you can do operations on, like equivalence transformations on, before you go to the numerical simulation. Here's an example in plant development where we have a Mathematica embedded language called CellZilla that expresses a whole model. This is a gene regulation network model called Wushel. The key gene is called Wushel, and it has these peaks, and it's something that we'd previously published on to describe real microscopy operations. And right there is a cut and paste symbolic expression of this model that you could put into our Mathematica package and run it. So this is a declarative model entirely in a computer algebra system. Here it is in Python instead, and it's a different model, but that's the same language. And here's the same kind of model. Actually, this is the Dynamical Grammars model now, used in a plant root simulation by Guy Josefan in 2009. Here's the actual cut and paste expression for a few of the rules in this model. It's got cell division, which are discrete events. It's got differential equations that you solve. They're intermixed between these discrete and continuous things. And so it's quite a powerful modeling language for developmental biology. Well, what's the general setting then? In a modeling language, I think the operator algebra formalism suffices to give you all the process models that you need. So you need, you have these general rewrite rules that turns before objects into after objects. They have operators that describe what's their semantics, and those operators obey an algebra. And it's all compositional. If you have, in various ways, for example, the operators add up when you have more rules. But the developmental biology and structural cellular biology really show you you need something more than just processes. You need sort of non-trivial objects, evolving shapes on which other processes happen. And for that, we need not just the verbs in a language, but the nouns of a language to be quite elaborate. And that sent me on quite a detour, which I'm going to drag you through a tiny bit into the types that you use to mathematically constrain what are the possible objects. So I suggest that type theory is going to come to the rescue. I guess I'm going to skip this slide because I'm going in a different direction now. So what are the process semantics is operators that basically act on giant probability distributions in a master equation. What about objects? Well, here's what people classically want in type theory for a programming language. They want sum and product types. They want function types. And then maybe they want a few more things too that allow you to restrict one type to another. And a lot of that is provided mathematically if you have a Cartesian closed category. That's kind of the goal of a lot of programming language theory research, is to provide CCCs for programming language semantics. And then you can do these type judgments like the thing showed on the left where you have type A and type B, then you can get a cross product type. And this is okay-ish for topological spaces. They exist. There's a nice correspondence between logic and type theory and category theory called the Curry-Howard-Lambeck correspondence. So there's a potentially deep mathematical framework, but there's also obstacles. For example, probabilistic programming doesn't really have a CCC. There's a couple of suggestions for that, how that could happen. And we need something like that for stochastic models in complex biological systems. So, what are we to do? So my suggestion here is to make a variant of type theory where we have, for each type, for example, two-dimensional manifold representing the surface of a cell, we have three things. We have a category whose objects are the elements of that type. That's the unusual part, actually. We have a type symbol tau for that type, which is an element of another category, which is the category, which is the type theory category, and that's the standard part, actually. So that in the Curry-Howard-Lambeck correspondence, one builds a category of types, which are objects, and the maps between them give you more terms that build more types, and so on. And finally, a sublanguage, which is the part that a practitioner modeler would be interested in. And so that's an eclectic type. It's a little bit more apparatus, and it should have types. It should have subtypes, and they should fall in some kind of type hierarchy that is. So what's new now about the subtype is that you're going to have some kind of relationship, like a functor, between a more special type and a more general type. And then you can keep on going with the hierarchy of types and have the idea of a type module in which you only specialize but not generalize. And when you want to generalize, then you make a new type module and put that into a curriculum of type modules. So, well, what could you do with all this? Here are the kind of types that I'd like to have for modeling languages. You can see deep in the middle there's manifolds and cell complexes and stratified spaces. There are processes, all the operators that move probabilities around on these things, so you need function spaces. There are expressions for abstract syntax trees on the other side. So that's the bottom part. Those are the things that you want when you're a practitioner in modeling. Along the top are the very general ideas, like a distance metric, a sigma algebra, a measure, a topology, and in between is unknown stuff. Every time I try to draw it, it gets huge. But you get things like topological vector spaces, and so a really intelligent mathematical modeling system would know all this stuff. So who else is working on this kind of thing? Well, for example, here's the Lean Math Lib that has just such a type hierarchy. This is the algebraic part of it, or an algebraic part of it. In this case, it's kind of upside down, I guess, from the other case. So very general ideas at the bottom, and specific usable things are at the top. Here's a similar thing from a workshop on defining computably definable function spaces for use in computer algebra systems like Mathematica from a few years ago. So the world of computer algebra and the world of automatic theorem verification are both facing the same problem. Let's hope they solve it. My plan is to approach it through these graphs and graph limits and so on. So, so there's a lot of work to be done there, but I think that's the process. That's the way to go beyond to have a whole family of languages of increasing capability for complex biological systems, and I say that as someone who's built such a family of languages, but much more simple ones so far, like the ones I showed, we need to keep on going. So let's imagine that you have many such modeling languages of varying capabilities. What's the, how would you access the right one? Well, you have some kind of cabinet that you put them all in that's very organized. And here's the proposal for the organization. It's some kind of cross product where we have different knowledge domains, you know, your science, your mathematics, your computational expertise, and if you're a modeler, then something in between. And then that, for all of those things, you're going to have objects, processes, and expressions. And then, so that's, you know, 4 times 3 possible nodes in a product graph. And then you multiply it by a huge potential hierarchy of different hierarchical relations, both compositional and specialization-like relations, and go down for each node. You go down as far as you need to in order to get the thing that can that has been done. Try to find something that has been done that is adequate to your biological problem. So, and here are the approaches, the formalizations that could apply to each of these things. Processes, operator algebra, objects, type theory, as I mentioned, and then, you know, formalization in science, description logics, perhaps things like the Go hierarchy, and so on. So I think I'm winding up, but so every node in this resulting hierarchy is going to be a modeling language. The modeling languages have mappings between them, like semantics, model reduction, you know, smart mappings that are undergirded by symbolic AI or by numerical machine learning AI or both, and that's where the intelligence goes. And then if you put it all together, then on the lower right corner here is the same, is the you know, domain map complete with the different kinds of mappings between them. You would do some modeling that had a mathematical semantics psi that addressed a scientific problem by way of the C, you know, problem expression mapping, and that finally could be implemented in terms of, you know, in something like you know, your favorite modeling language backend could be Kibana running on an exascale computer, for example, with machine learning algorithms running in it to find model reductions while it runs. And that's the big vision. And I hope to find, if you're at all interested, I hope to maybe hear from you so that I can figure out how to organize some kind of approach on that. So what I hope to have described to you, at least, maybe not convinced you of every point, is that we can use machine learning to bridge spatiotemporal scales. Here I gave you the examples of dynamic Boltzmann distributions and graph prolongation convolutional neural networks for you know, real biology problems in synapses and in plant cells. Then a big leap forward in expressive power for coarser scale modeling, and model reduction tends to go coarser scale, and that's dynamical graph grammars, which are well defined and have a semantics, and you can create simulation engines for them. And then and much more speculatively, of course, is the steps towards this integrative conceptual architecture, which would be how you could really leverage all of these things in order to make a sort of artificial intelligence of mathematical modeling of complex scientific situations. And with that, I think I gave you all the output that I was hoping to output, and I'm happy to take any questions.
Very good. Thanks, Eric. Yeah, really a very inspiring talk, and biology is just such a wealth of concepts and structures that we need to describe. We'll probably be doing this forever, long after we've solved physics. So, okay, I'll open it up to questions. I'll propose a question after. Are there questions from the audience? Let me throw out again. It's just, you know, so many possibilities, so many directions. There's one connection maybe we could I can mention with I mean, obviously a huge and long-standing field, but one of our postdocs, Michael Simkin here, I hope he's here listening. So you know, the whole, you know, taking taking basically discrete or, I mean, a discrete structure such as a graph, or a graph with state, and developing, a stochastic process of how it evolves, and then perhaps taking a limit, getting a much simpler equation that obviously can be used in a huge number of domains. And he's been using that in combinatorics. There's apparently all long-standing method in combinatorics of the differential equation method to in his own recent work, for example, getting bounds on the 8 queens and the n queens problem of putting queens on the chessboard. I wonder, Michael, if you're there, does any of, because again, if you look past the differences in language and formalism, at least the starting points have much in common. Does any of what he said resonate with you in any way?
Well, I think, yeah, hi. I think that much of what was said resonates with me. I mean, there certainly were a lot of similarities that are between like what I was thinking about and what was said here. I definitely need to think about this more and see.
Right.
Yeah.
Right. And then Eric, is there so if we just took like your starting point, we have a graph process, we have a stochastic graph grammar. Is there an actual rigorous formulation of that even like basic statements have been proven?
Well, it's the master equation in my way of thinking about it.
Yeah, sure, that's right. I mean, physics, again, I'm on some level familiar, but have mathematicians made the basic definitions to their satisfaction that could be served as like a foundation, for.
I don't think so. I don't know of a lot of other work that would put graph rewrites into continuous time using the master equation, except for the 2 things that I mentioned, which is, my work here, and then the double push out framework here, which does go back quite a ways and is not, it's discrete in time, and not especially probabilistic, but that's
Okay.
the other place to look for
Right.
existing
Well, again,
foundations.
it seems very fundamental on both sides. So perhaps there's some connection to be, some discussion to be made.
I think so. Yeah, I'm eager to find out about this combinatorial work.
I'll be happy to talk about it.
Good. Yeah.
Other questions? Well, okay. I mean, to throw out general questions. I mean, yeah, obviously there's this problem of you have a very, very complex simulation, and you need to find a sensible reduced model. I mean,
Right.
it's such a broad and wide-ranging problem. I mean, let's see. I guess a general question I could throw out are cases where, in biology, people could say, not that the problem is solved, but that there's like here's an example that really kind of a lot of people rely on. Here's a reduced model. I mean, again, at some level that's been done a lot, but one which, let's say, machine learning or more modern tools had an essential role in formulating.
Yeah, that makes it harder, of course. I mean, my vision for the Tacoma architecture is that if you didn't know the right math already, it could do some pattern recognition for you on your input model and say, well, I don't know, but 2/3 of your model would fit into this framework where I can do an analysis.
Right. Again, you're talking about making things easier for the researchers, and I'm asking for like the case where one, a success.
Yes.
I mean,
right. So, I mean, the standard things in well, a standard approach is to create a partial create a reaction diffusion kind of model, then make a continuum version of it to a PDE where you replace all of space with diffusion with diffusion, and then to do a linearized perturbation theory of that thing in order to find the fastest growing modes in a pattern formation. I would say that is like one of the most common ways of making your living if you're a 1980s theoretical biologist, and that could be automated. Absolutely, I see no reason why not. Then more recent variations where the spatial signaling is not diffusion, it is these highly nonlinear signal transduction things. You could even make and people have made a continuum model like of our pattern formation in the shoot apical meristem of the plant for where the next flowers go. People have made a continuous version of that where the differential operator turns into a fourth order nonlinear kind of thing. Ordinarily, you'd be scared of it. You wouldn't think it was going to be stable, but because of the biology, it is, and you could do kind of the same things. But then it suggests that new analyses could be developed as well. So that's a good starting point. Then of course for stochastic models, the first thing everybody does is something like mean field theory, ignore all correlations. But then there are slightly more sophisticated standard things that people do. And there's the Kirkwood superposition approximation, for example, which is off the shelf of the StatMech kind of methods that has been used by Miney and Baker in a paper that I kind of liked. That is, again, on the road to going from a stochastic model to a PDE that you could then do more normal, more conventional analyses on. So I think what we can get, I think there are lots of individual examples, and what's exciting about trying to automate them, besides the fact that more people could use them, is that you could then also find their validity bounds in two ways. One is what's provable, and the other is can you use the machine learning to observe doing it many times and predict when it's actually valid outside of the provable range. And so yeah, these nuggets of known analyses are valuable, and we should try to express them symbolically.
Okay, yeah.
So those are two of them.
Right.
I'm sure if I sat and thought, I could think of some more standard analysis techniques. I mean, another one is if you have a gene regulation network, the first approximation that a lot of people do just automatically is they simplify it to a Hill function. They simplify a lot of things to a Hill function. You can find out when is that valid, but you could also, make the machine that does it and find out not when is it analytically valid, but when is it observed to be valid in a bunch of simulations. Because I think it's incorrect in a lot of cases that really you need a different ratio of polynomials model that reflects more structure of your gene regulation network, but it often doesn't get you into too much trouble.
Okay, yeah, great. More questions?
All right. Well, thank you. If anybody is interested in helping, let me know.
Great. Okay. Well, we'll certainly continue the discussion. Okay. So thanks everybody. And next week we'll have Yang Hui, who will talk about machine learning, big data for universes. So until then.
Hey, Michael, if we want to help Eric, what do we do? Do we just email Eric, or what do we do?
Yeah, I'll post his. I mean, he's at UCI. You can look him up, but I'll post his address, and certainly I'm sure he'd be happy for us to contact him.
Yeah, thanks so much. This is like a lot for us to digest.
Yeah,
Michael and some of us, we've been talking about this for a while, and then wow, it's like a drinking from a fire hose. So thank you.
Okay, thank you. Okay. See you later. Bye.
Bye.
All right. Thanks again for the invitation. See you later.
Yeah, that was good. They really did give a good sense of what kind of things you do. And I think there are connections there with things people I'm talking to are interested in.
Great.
Yeah.
See you later.
Yeah.